Thank you, Jonathan, for the introduction. I apologize because of the time difference. I wasn't able to follow most of the talks. I'm practically 7 hours behind. Uh, but my presentation will have little to do with protein dynamics, but rather putting everything in the context of drug discovery. So I'm going to give you a quick tour of the power of pretty much data science and a little bit of AI or machine learning, as we prefer to call it, in the process of drug discovery. So very briefly, I will tell you a little bit about, I'll start with targets, then I'll go to diseases, touch on drugs, and then I'll finally converge on the almost druggable genome, which the only reason it lost the quotation marks is because I forgot to put them. So on targets, we have developed a comprehensive list of drug targets back in 2017, and we've been slowly updating them. So I'll share the slides with everyone who's interested, but essentially these are links to drug targets for drugs approved in 2018, 19, and 20, and they were also published as one-page little updates in drug discovery. So I'll really encourage you to check the list of what we currently call TICLIN. But the more context, that the bigger landmark publication related to this project called Illuminating Druggable Genome was a year later in the same journal. And essentially, this program has three major experimental groups, which are divided between UNC and UCSF. There is an outreach component and a knowledge management center, which I share with Avi Mayan. And then there are also cutting edge informatics technologies grants, which focus on developing tools that feed into Illuminating the Druggable Genome. So part of this initiative, we were lucky enough to be able to develop an interface that we call PHAROS. And what feeds PHAROS is a database called Target Central, and this illustrates briefly the process of assembling Drug Central, which essentially means we start with a lot of information from large-scale experiments and publications. We had to develop, in addition to existing ontologies such as Mondo and Diseases and Uberon, we have a drug target ontology from Stefan Schürer's group. We also incorporate chemocentric databases such as Campbell, Drug Central. I'll touch upon that later. And then all of this gets collated into Target Central. And this is available in machine learning ready format. So there is an adapter that you can use with Python or if you prefer R, but we are mostly focused on the Python version at the moment. So you can take out the data and then dynamically or statically assemble your preferred machine learning models. And I'll briefly touch upon that as well. We have lots of collaborators. You can visit commonfund.nih.gov for information about Common Fund. And of course, our resource is druggablegenome.net, and PHAROS is at pharos.nih.gov. And these are some of the data sets that we are incorporating, and you could insert your own data to this, of course, but essentially what we try to do is capture information related to structure and function, pathways, expression data under different conditions, as well as additional information related to diseases, phenotypes. So all of that we try to bundle together. We have proposed a knowledge-based classification for proteins. I don't have enough time to go into details. 30 minutes goes very fast, but if you look at 3 o'clock, that is roughly the approved drugs target. So these are proteins that we call TICLIN or mode of action proteins. And then on the upper side is TChem, which are proteins that are modulated by small molecule chemicals. And the latest updates puts those numbers at about 9% for TChem, so a solid increase, almost more than 2%. And then about 10% more compared to the initial set. So out of 600, we're about 670 now when it comes to T-CLIN. So what's shocking about this is that about 1/3 of the proteome, and that number is slowly decreasing, are proteins that are poorly studied or understudied proteins. They lack enough antibodies annotation, they lack publications, etc. So that's what we call T-dark. And then about half the proteome is what we call T-bio, meaning we understand some of the biological function, but we do not have small molecules or drugs to modulate them. Why should we care about T-dark? This just illustrates how a completely dark protein, so these are some of the most famous examples. Dark protein in 1995, by 2014, there was a drug on the market. And you can see that the pattern of their best study publication was either receptor deorphanization or a strong disease association study. So on average, it would take between 15 to 20 years for a dark target to become T-CLIN. And this is a more recent effort that I'm doing together with John Overington, and it's essentially taking the first year of publication for a protein out of UniProt. This is, of course, human proteins only. And we're tracking publications from 1964 to 2007. And you can see that although there was some sort of an initial interest in proteins related to the immune response, that interest sort of dwindled over the years. So in fact, the fastest growing in terms of publications were nuclear hormone receptors, signaling proteins like cytokines, and then kinases, surprisingly enough. And then the slowest one to date is olfactory GPCRs. If I understand correctly, that has to do with the difficulty of studying them in experimental conditions. So the pitch to go visit pharos.nih.gov, you can query by ligands, you can even do substructure search as of July release. And if you want to implement this internally, there are certain GitHub elements that you can access. So the quick take-home message here is that there is a knowledge deficit. About 3% of the proteome is currently targeted by drugs, and about 30% of the proteins are understudied. So going to diseases, this is the tool that we use primarily. It comes from Lars Jensen and his group in Denmark, and it essentially has three channels: text mining, knowledge, and experiments. On the experimental side, we just recently contributed through a paper in bioinformatics a tool that we call TIGA, which stands for Target Elimination by GWAS Analytics. It literally adds an additional layer that looks at relative citation ratio on top of GWAS scores. Other than that, we rely heavily on text mining, and after evaluating multiple transformers, Lars has decided that BioBERT fits best when it comes to text mining, full text out of PubMed Central. So this resource, full text mining, about 2 million papers, it's updated every week. I encourage you to go visit it. So why do we care about diseases? Well, this just illustrates the knowledge gap between the number of drugs that are on the market. Pardon me. This is essentially orphan drugs approved in Europe and in the US, and a number of publications for proteins that are associated with rare diseases, which literally goes by 3 orders of magnitude higher every year. So there clearly is a gap in translation. And part of that gap is we don't even know how many rare diseases there are. We wanted this to be the title of the paper. The title is much more bland. It's just how many rare diseases are there. But the reality is, if you do not count rare diseases, then rare disease patients do not count. So what we did is to take a lot of resources that are publicly available, of course. And if you put them in an ontology and then look at what's called leaf terms, meaning you can no longer go below in the hierarchy, you end up with about 10,400 rare diseases according to the multiple sources. And we made a pretty decent effort to reconcile the different nomenclatures. And of course, that number continues to change because of another phenomenon called the ultra rare disease patients, which are like one patient on the globe. So part of the question and what we're trying to answer is, can we look at this type of relationship between diseases? For example, is it a disease causing germline mutation or loss of function or gain of function? And then try to color that in terms of the relationship between proteins and diseases. And again, using the rare diseases resources plus Lars Jensen's, we concluded that, contrary to what's claimed in the literature, a little bit under 5,600 proteins have relevant verifiable associations to disease information. And of course, we have an urgent need to improve that type of relationship. Just because a protein is overexpressed in a particular disease doesn't mean it has any relevance therapeutically. So that's another thing that we could talk about. But one of the questions that's been often asked is, can we use animal models? So one of the other common fund projects is the International Mouse Phenotype Consortium. And out of the IMPC, there's about 7,000 human genes that have been associated with diseases. And so the question that we can ask is, can we look at the phenotyped mice and try to establish a relationship? And there's an interesting cluster, which I colored in gray all the way to dark, and there's reasons for that. But the short of it is there's about 1,400 genes and phenotype mice that do not show relevant phenotypes in a battery of more than 500 tests that they run. So we think that what would contribute to this is rare diseases. And one way to look at this more carefully is to ask the question, how many of these rare diseases have gain of function? And the answer is about 177, so surprisingly not much that are annotated in the literature. And then the other one, of course, is the loss of function. And these are, of course, more almost 1,000, so 6 times more diseases. But at the end of the day, what we're trying to establish is the impact that this could have into relating mouse phenotype with human disease relevance or symptom, and then to try to establish whether this could serve as models for therapeutic effect. So why do we care about this function prioritization? Is because if you start with monogenic diseases, at least you understand which is likely to be your target. But the other part of what we're dealing with is in therapeutics, about 90% of the drugs that are out there are either antagonists or inhibitors. So it stands to reason to assume that they effectively block some sort of gain of function that's related to that disease. Yet this is very poorly annotated in the literature. But in principle, we think that as we increase our understanding in this area, we could probably develop new rational strategies for therapeutics. So this is an AI conference. I will briefly mention, I hesitated whether to put these slides in because it's in the interest of time, but essentially we have built a rather complex system where we take the data that I mentioned in Pharos. It gets into a database which is then allowed to automatically build its own models, either taking the phenotype series from OMIM or a phenotype series from the IMPC dataset, the mouse models, or you can come up with your own list of files. And then following a knowledge graph embedding, it adds dynamic features to the set. These are static features on the left top. And then we use XGBoost to build the models. So out of that, one of the lessons that I learned is do not trust machine learning. So the first attempt I did to model type 2 diabetes was taking data from OMIM. And what I got out sort of made sense. Almost 90% of the proteins predicted by the model are expressed in the pancreas, even in the beta-Langerhans cell. If you look at the features, the top one is hemoferritin, which mutates in a disease called hemochromatosis. Most patients with hemochromatosis develop type 2 diabetes. So I sent this to Mark McCarthy, who at the time was in charge of the type 2 diabetes consortium. And well, it turns out that what I learned from him is that the model I built was essentially useless. They could not see anything in the test set or the training set. And then they even questioned the input that we used. So the short version is they sent us a completely different set of genes, and the model that we built, even though computational speaking, it looks very similar, area under the curve is similar, the features gained are very different. And you can immediately see just by looking at even the top 24 genes that the majority of them already have some sort of GWAS association with type 2 diabetes. Even 2 of the top 4 nodes by feature have relevant type 2 diabetes association. So very, very different impact of the model. And then what we did is we validated this with additional data from the IMPC dataset. So there are 5 different phenotypes, and this is the number of genes that is associated with that. And of course, with IMPC, the advantage is that you can use everything else as a negative set because they are not relevant to type 2 diabetes. So then you can combine these models with a human model, and then you can come up with a much better list of genes that might be relevant to type 2 diabetes. So just to quickly summarize this particular section, you need experts to talk to. So without an expert, I've thought that the initial model that we built made pretty good sense. At least it made sense to me, and I'm not a diabetes expert. So talking to experts was important. Equally important, between the 2 models, computers could not distinguish which one has more value. So you need lateral validation, and lateral validation is what we got out of the IMPC dataset. So the short story here is that I think dark genome research can benefit rare and common diseases with information that you can extract from patients and mice. So very briefly, I'll go on to drugs. In drugs, we have developed this resource called Drug Central. This is by far the most accessed resource that we have. Just 1 year alone, we have more than 100,000 users. And we recently just added sex-based differentiation of side effects, extracting data from FIRES, the Federal Adverse Events Reporting System. This is just to give you an idea of how complex the database is. I won't even go into the details of this. It's just the schema, but we pretty much track information from multiple sources related to diseases, drug targets, side effects, chemical structures, etc. So the most recent, the only AI slide that I have left is that we built this platform called Redial 2020. It was, of course, meant as a joke, but essentially we took data from the NCAT's COVID-19 portal, and then we built a platform that currently has 11 assays on top of which we built machine learning models. So you can go to Drug Central/Redial, type in your favorite chemical or drug or PubChem ID, and it will predict 12 different activities. We added the sigma receptor just recently. And this is published in Nature Machine Intelligence, came out last month. So going back to the almost druggable genome, how do we fit all these things together? Well, it all came from a challenge from Al Edwards. Al Edwards, who leads the Structural Genomics Consortium, is putting together something called CASH, Critical Assessment of Computation Hit Finding Experiments. And it's co-organized by SGC together with scientists from Bayer, Google, and the EBI. And the purpose is to try to understand how many proteins could we access through virtual screening And part of his questions that he addressed to me were, can you come up with a list of proteins that have structures, no ligand, structure with bound ligand, structure, ligands, and SAR, and then a close structural homologue without ligands. So very briefly, structure, no ligand, and structure with bound ligand. This is an analysis done by SGC's Matthew Shapira. Essentially, these are the numbers stratified by target development level. So if you focus on those that lack chemical matter, which are T-bio and T-dark, you end up with about 700 proteins that we think could be immediately ligandable, meaning they seem to have a ligand bound, but probably not a very potent one, or there is a PDB which might have a cavity or even an endogenous ligand, but it wouldn't be on these two other lists. So that would be one way to look at it. The other way to look at it is to ask the question, well, how many proteins have been tackled by medicinal chemists? And the answer is about 2,600, which have unique UniProt IDs. I sort of stratified them by the number of active compounds that are associated with each protein. So significant SAR all the way to no SAR, you only have one compound. So in the no SAR, T-bio, T-dark, you have 115 proteins. So these have ligands, but they're not very potent. So again, this could be also immediately ligandable. When you look at the intersection between these, if you focus on the no SAR and no chemistry, you end up with about 700 proteins that have a good 3D structure, but again, no chemical probes associated with them. So this one would deserve more attention. Essentially, what we did was to go through using the MM6 algorithm. We went to do a homology modeling sort of automated system, or we think we could build homology models. So we looked at similarity between all the UniProt proteins, so 20,000 by 20,000, and then 20,000 UniProts by all of 600,000 proteins in PDB. And the question then was, is one of the pairs already with a 3D structure, or is one of the pairs already with the TCLIN or TChem? So the TCLIN and TChem ligandable no PDB end up in this category. Those that are ligandable, which suggests that you could actually quickly build virtual screening models, are here. You could do 3D models likely, and that likely I put 60% protein identity, which shows already very high, and then between 30 to 60%. So all told, you might be looking at another 2,600 proteins that could be immediately ligandable. So this could literally double what we know out of medicinal chemistry, which were the other 2,600 that I mentioned earlier. The other question that one can ask is how much information do we have in disease space? So in disease space, you can literally look at gene ontology terms, IMPC knockout mice, GWAS traits, rare diseases, common diseases, etc. And this again gives you an idea. If you're going to prioritize based on disease associations, you should probably reduce the initial set to about 600 proteins, which are ligandable with 3DB, 144, etc. So all of these, I think, are really good ways to prioritize proteins. So to provide a last answer to the CASH team, we think there's 700 ligandable proteins that should be considered for the CASH competition. There's 180 that I think would be of immediate interest. And then perhaps these T-bio, T-dark proteins with no SAR, which do suggest that you could build and optimize the probes that have already been disclosed. What I intended to do in preparation for today was to enrich the analysis with PFAM domains. I almost finished the work, but I got stuck in the last 3 days and I couldn't make progress. So I apologize for that. I was going to tie in together all this information really nicely, but I just didn't have enough time. So the final message, the truth is already here, it's just not evenly distributed. The really hard part of any AI initiative is to capture what's called a ground truth, what we really, really know, and not what you think you know. And then it's also important to talk to domain experts Otherwise, you can go in a wild goose chase. So with that, I hope I'm on time. And again, I'll be happy to share the slides and answer questions.